Okay. And now I will introduce our first speaker, Lucía Moreyra, who will be presenting their work on the origin and diversification of Cirsium.
Hi, everyone. Can you see me and hear me correctly? Okay, I will share my screen. Let me see. How can I do this? Okay, so thank you so much for that introduction. I'm going to start now. So as mentioned before, today I'm going to talk about Cirsium, which is a thistle genus. This work was part of my PhD thesis, and it was published this last January in Molecular Phylogenetics and Evolution. So I'm going to start with a little bit of context. As you know, within the Compositae family, we can find a very wide variety and diverse of species, and usually this diversity is grouped into tribes. One of these tribes is a very diverse one, is the Tricarduea, which is, as you can see here, let me see if I can, here. You can see there's a variety of different subtribes, and my focus of study is this subtribe here, which is Subtricarduinae. So within this Subtricarduinae, there is a very wide group. Let me see, 1 minute. Okay. Within this subtribe, we can find a very wide and diverse group, which is the Cardocirsium group. It has a different amount of genus with different amount of species and different, very different richness in the diversity. For example, we have around 6 genus with between 1 and 20 species, and the rest of the diversity is divided between Cardos and Cirsium. And actually, as you can see in this pie chart, Cirsium has accounts for around 75% of the diversity in this genus, in this group, sorry. So Cirsium has around 450 species. As you can see, it's distributed mainly across all North Hemisphere, and its distribution is very different. It has a very different points of diversity. Mainly, we can find species in Southwest Asia, and it has 2 other centers of diversity, which is North America and Japan. So regarding how this genus is classified, mainly there are different classifications, but the most accepted, at least now, is this classification, which divides Cirsium in 2 different subgenres. One is subgenus Cirsium, which is comprised. It comprises a lot of species. Mainly. Sorry. Sorry. I thought someone talked. No. Okay. So within Cirsium, the subgenus Cirsium, we find a lot of species. Mainly, they are distributed across North America, but they are also distributed across all North Hemisphere. These species are both mesophiles and xerophiles, but subgenus Ophiulepis is mainly distributed across this area you can see in here, and this genus has mainly xerophyte species. So, as I mentioned before, within Cirsium subgenus Cirsium, there are 2 main centers of diversity. One of them is this center you can see in here in North America. This group has around 100 species, and it's mainly the only one of the groups around Cirsium that has been studied in terms of biogeographic history. And actually, there is this work led by Carolina Seriskalki. I'm not sure if I'm pronouncing it correctly, but in this work, they found that probably all species of Cirsium across North America, they are probably the result of a very rapid adaptive radiation, and probably it originated from a single dispersal event from probably some part of Asia through Beringia. So the other main center of diversity, as I mentioned before, is Japan, which is quite surprising because it has around 80 species, at least accepted now. And it's quite curious because Japan has around 20 times less land than North America, and we can find a vast variety, a vast amount of species here. So this group actually particularly has never been studied or included in any molecular studies. So we were quite excited to include species from Cirsium from Japan, so we can try to know this group a little bit more. So our main objective for this work was to establish this possible origin of Cirsium and try to know a bit more about the possible dispersion patterns, especially not only the whole genus, but also to try to understand how the two subgenera colonize the different areas. And maybe we can tell a bit more about the biogeographic history. So within all the studies, including Cirsium, they have two things in common. Mainly, they have two limitations. One was incomplete sampling. They were only focused in one certain area of the world. They were never including all species of Cirsium. And also, they have quite low phylogenetic resolution because Cirsium has a lot of species that originated in a very short span of time. So it's very difficult to obtain a very supported phylogeny and very defined. So for this, we try to include all samples we could graft, and also we apply the next generation sequencing, especially using the HiSeq approach. So in total, we gathered 266 species of Cirsium, which accounts for around 60% of the genus. And then we included other species, which were from the rest of the Cardocirsium group, the subtribe, and also the A group. So I will go very quick about what we did in the lab. The sample processing was, of course, extracting DNA. We did library prep. We applied the target capture protocol using the composite 1061, and then we proceed to sequencing. And what we obtain from these raw sequences, we apply the HiPiper pipeline, and we use also the supercontic option to recover not only exonic sequences, but also intronic. And in total, we obtained 350 loci, which is quite low if we compare with the 1061 loci that we should expect from the kit we use, but we can discuss later about why this is very low. But in total, we had these sequences. So with this data, we build two types of phylogenies. We applied the concatenation approach and also the coalescence approach. We try to also compare and be sure that everything was quite congruent between the both topologies, and more or less they were quite congruent. So we proceed later with the concatenated phylogeny. We dated that phylogeny. We delimited the areas. This is for doing the biogeographical inference. Then we build a matrix with all the distribution for each one of the species. We apply the biogeovirs package in R, and then we apply also a biogeographic stochastic mapping protocol to try to understand a bit more where these dispersed were coming from and where these species were distributed to. So the areas we define were these you can see here, which includes mainly all North Hemisphere, and also we include this area here because we had some genera within the Cardocirsium group that were also endemic from this area. So, but Cirsium is mainly distributed across these areas we can see in here, and is the main focus and is the main areas I'm going to explain now. So regarding the results, the first thing we find out is that Cirsium probably originated around 9 million years ago, where the temperatures in the global tendency for the temperatures were to cooling down. And we also find out that, sorry, you can see this part because I'm seeing this. Let me check. Okay, now. We also did a diversification study, and what we found out is that the diversification rate in Cirsium trifold around 4.6 million years ago. So this happened during the early Pliocene, and we are going to talk a bit more later about this, but these were the main result regarding when Cirsium originated and when things happen in this radiation of Cirsium. So regarding where it originated, we found that the ancestral distribution for the whole genus was probably in the Western Palearctic region. And we find out also that both subgenera originated mostly in parallel around 7 million years ago. And what regarding where each of these subgenera originated, subgenus Lophiolepis probably originated here in this area, which is the Mediterranean basin. And from here, we saw that a dispersal to Southwest Asia happened, and this is coincidental with this moment where the diversification in Cirsium trifold. So as you can see here, a lot of species originated in a short span of time. This diversification started happening very quickly in this early Pliocene. And we are going to talk a bit more later about what could have caused this pickup in the diversification rate. So from this South Central Asia diversification, we also saw that later on, dispersions to Central Asia and back to the Mediterranean also happen in this subgenus. But this subgenus mainly kept diversifying in this area that we can see here. Regarding subgenus Cirsium, probably the ancestral area was around also the Western Palearctic, the same as the genus. And our results show that different dispersals occur, especially from this ancestral area to Central Asia, and also from probably also this area to in parallel to Southwest Asia and also to East Asia. So from here also we saw a dispersion to the Macaronesia, which originated also a species in there. And also we saw another dispersion in parallel to Central Asia. And this dispersion happened just before this pickup in the diversification rate in Cirsium. So from here, once it reaches Central Asia, we found out that these several dispersals happen, especially to different parts, the different areas of Asia, especially Japan. A single dispersal from this central area to Japan happened around 2.4 million years ago, which is the beginning of the Pliocene. And this dispersal originated this vast majority of species that we see in Japan. And actually, this is a map showing the distribution of several species of Cirsium. Each one of the circles indicates the distribution, of different species. And these letters also indicate different species that had a very narrow distribution. And these species actually are very adapted and thrive in different habitats. For example, they are mainly adapted to much wetter habitats than the rest of the genus, and also they have these short short distributions. So one of our main hypotheses is probably a niche shift probably happened after this colonization of this area, and probably this fluctuation, because this happened during the Pleistocene. These different cycles of the Pleistocene probably connected and disconnected different areas of the Japanese archipelago, and also connected not only these islands, but also with increase and decrease this connection with the mainland. So probably our main hypothesis is probably both allopatric and ecological speciation is what gave origin to these very wide adaptations that we can see in Cirsium and all these different species in a very short amount of time. So regarding the other main radiation that we know, which is the North American one, what we found is that probably a single dispersal also from Central Asia to Beringia most probably reached North America and originated all these species that we can see here. And this dispersal our data is showing that probably occurred around 1.3 million years ago, which is also during the Pleistocene, and our results are according to this fantastic work regarding this biogeographic history of the North American thistles. And we also believe that probably allopatric and ecological speciation happened here, which was probably it developed after these cycles of the Pleistocene, a lot of refugia, different connections and disconnection, and different new habitats to colonize. So regarding our biogeographical stochastic mapping, what we found is that the main areas from where this is showing how the areas connected across the whole history of Cirsium, and we can see that this Mediterranean basin and this Southwest Asia were the main origin of this dispersal, and that dispersals were going in this direction to the east, reaching Central Asia as well. So our hypothesis is that because all these different pickups in the diversification occurred during the early Miocene, and also the main jumps to Asia occurred during the end of the Miocene, probably here, the close of the Parathetic Sea and also the conformation of the Mediterranean basin, this increased not only the connection between the different areas, allowing these especially certified species to disperse, but also in this area, for example, in Central Asia, the monsoon system started to pick up a lot, and it increased a lot of the aridification in these areas, probably also benefit the thrive of these xerophytic species. So this is our main results in a very summarized way, and this is mainly the end of this thorny tale so far. So thank you so much, and I hope I didn't overdo the time. Thank you so much.
Thank you very much for a great talk.
Oh, thank you.
Yeah, very interesting.
Let me see.
Do people have questions? If you'd like to just unmute yourself or put a question in the chat, whatever you would like.
Let me see if I can hear you better. Yes. Okay. Yeah.
Christoph.
Or you're in mute, Christoph.
So now, thank you for your nice talk.
Oh,
So
thank you.
I was just wondering about how you tackle the problem with polyploids in Cirsium and with the reticulate evolution. I think I remember that there are many polyploids in Cirsium.
Yeah, there are many polyploids in Cirsium. And actually, when I started explaining a bit about the lossy recovery we had, actually we, and someone is, at least is asking the same in the chat, actually, we recovered, we have a lot of paralog issues because, yeah, we didn't exactly apply any protocol to account for this polyploid, but what we find out is that these not only polyploids, but we also have a lot of allopolyploids, which is giving a lot of issues regarding this paralog recovery. And when we try to build phylogenies, we discovered that a lot of genus were, a lot of different loci were recovered as paralogs that were actually orthologs, not paralogs, and sometimes they were just putative paralogs because they were just copies of the different polyploid species. So we end up just picking up the loci that had non-paralog warnings, and we also built phylogenies for 500 different loci, including the paralog copy and the ortholog copy, to assess and be sure that these paralog warnings were being real, these putative paralogs that they were, that the program was picking up, they were actually paralogs. And that's how we end up selecting this loci to build that phylogeny. But it was really, really tricky. And we spent like 3 months, something like that, trying to figure out what was happening. Because at the first we obtained, I think the first tree we made, we obtained, for example, all the bootstrap values and the LPP values were 0. And we couldn't understand what was happening. And then I understood what was happening because on our reference, we tried to polish a bit the reference, and I include one species from one of the subgenus in the reference to have better recovery. But it was at some point the software was making a lot of mistakes trying to figure out which one were paralogs and orthologs. So I think there are better methods to overcome these polyploids issues, but we couldn't find a better solution actually for doing that.
How many species in your study were diploids and how many were polyploids?
That's the main other issue, is that we don't know actually exactly how many there were. Yeah, because we had a lot of course, a lot of collaborators that send us a lot of samples, and sometimes they knew and sometimes they didn't. So we try other pipelines also that can tell you with your HiSET data, they can tell you if they are polyploids or mainly if they are diploids or not. But they were really working very well because I had polyploids and I tried, and the pipeline was telling me, no, these are diploids. And I was like, no, these are not diploids. So we gave up a bit with that issue. We are trying to figure out how to overcome that, because with other groups, we are having the same issue. We really don't know how to solve it right now. So yeah, that's a great question. Thank you.
Yes, a question in the chat.
Okay, no, it's a great question for Japan. So the question is if we found out that all came from a very single radiation, of there were independent colonizations. We found out that, for example, we only have around 45 species, I think, from Japan. We didn't have the 80 species, but we found out that the main majority of the species, like 40 of them, they were the result of a single event of colonization, and they were all forming a monophyletic group. And then we found out a few independent colonizations, mainly from East Asia, but they were like independent events, and also there were species that you could find both in Japan and also East Asia. So this is the main result. The species can also reach after more recently they arrived to Japan, but the main radiation it's probably originated from a single dispersal event. Yeah, they are all monophyletic. Yeah.
Okay, we should go on to our next talk. So thank you again, Lucia, for a great talk.
Thank you.
And we can
Thank you for
have
this
more
opportunity.
questions at the end if people would like. So our next talk is from Vinicius Bueno, and who will present their work and their experience working on the tribe Neurolaeneae.
I will share. Thank you, Abigail, for introducing me. Do you listen me good? Everyone seen the share? Okay.
Looks good.
Thank you. Hello, everyone. My name is Vinicius Bueno. I am a scientist from Brazil, and I'm a person with a disability, because the title is Beyond Yellow Daisies, Dealing with a Neurolaeneae Systematics as a person with a disability. So Neurolaeneae was described in 1927 for Rydberg, and in 1973 Robinson Brattle transfers Neurolaeneae schistocarpa for to Senecio, and the tribe is considered a synonym. In 1981, Neurolaeneae was proposed for Harold Robinson with Brasilia, Callia, Green Maniella, Neurolaena, Stauroclamis, Tetrachiron, Chileropaps, and Unxia. Today, Brasilia and Stauroclamis are synonyms of Callia. And Schistocarpa was moved for Gallinsogini. In 2002, Panero and Funky proposed Neurolaene for treated tribe level, closer to Bai, Kenakchid, and Tagechi in the phylogenetic tree. Panero in 2007 described the morphology of the tribe, proposed 5 genera: Callia, Enidra, Green Maniella, Eptanthus, e Neurolaena. Panero described the morphology of the 5 genera also, and he proposed the number of the species approximately 150 species. So in 2019 or 20, in the papers of Mendel and Susanna et al., Neurolaene closer to Coreopside and Eliante. And Neurolaene is defined with 153 species. So I will show the most important researchers. Rydberg, De Candolle, and Baker was the more prolific publisher of species, describe species, new species for Neurolaene. Gardner and Snow. Snow provided the first and unique phylogeny, systematic review of Enidra, the genus Enidra. So more researchers. Turner, the most important researcher of is about Neurolaena, the genus. Panero, I mentioned it. Burhidi revised the Eptanthus, the genus Eptanthus. Harold Robinson and Prusk. Prusk I consider the most important researcher with about Callia. He studied a lot for more than 40 years. And Neurolaene has more than 350 names. And here in this graphic, we can see the impact of Baker in the Flora Brasiliensis, one great work about the flora from Brazil, where I live. And here we see the start of the impact of Turner and Prusk and Robinson also, when they started work with Neurolaene and publish Neurolaene names. Well, my first paper about Neurolaene, it's an overview I did with Morgan Gostel and Gustavo Heide as a part of my doctorate thesis. And we provided the first map of a distribution of Neurolaene. And I say special thanks for Bonifacino about this map. He teach me how I generate this map. And today we have, I consider we have 192 species of Neurolaene. Brazil is the country with more number of species, 100 for 3 genera, and 3 other countries have 3 genera: Venezuela, Colombia, Bolivia, in the South America. In the Central America, in the Caribbean, we have Mexico with 21 species, and Cuba also have 3 genera, and Jamaica. It's curious, Jamaica has 1 species of Callia, 1 species of Enidra, and 1 species of Neurolaena, one of them. In Africa, we have just two species naturally occur in the continent, two species of Anhydra, and one introduced species, one in the Democratic Republic of Congo, one Calea verticifolia, the species introduced. And in Asia, we have just Anhydra fluctans. In Australia, we have Anhydra wolves. So, about the genera, Neurolaena has 14 species just in neotropics, since in the north of Bolivia, from the center east of the Mexico, and all the countries of the Caribbean with Neurolaena lobata. The Eptanthus is a 7 species endemic of the Cuba. Eptanthus are very small plants with up to 10 centimeters, and I'm sorry, I forget to say about the morphology of Neurolaena. Neurolaena has paniculate capitulescence and pappus, elements of the pappus as bristles. And Eptanthus has pappus scales with a fimbriate margin, very fimbriate, you can see in this figure. Anhydra has 5 species, is the unique genus occur in all the world except Europe. And the morphology is very, very unusual, with a cupulate involucre, and the pappus is absent. Greenmaniella is endemic of the Mexico, has just 1 species. This species is characterized by radiated capitula and a shallow crown, very reduced pappus. In Calea, almost 90% of the species of the Neurolaena. Calea has 165 species. Characterized that all these species are characterized by a pappus scale without a fimbriate margin. And Calea can show different habits since 10 centimeters to 5 meters or 7 meters when the vine species. Brazil is the richness center with 164 endemic. We have 96 species of Calea, 3 Anhydra, and 1 Neurolaena. In Minas Gerais, the state I live in, has the most of the number of species of Neurolaena on the world, because just this state has more than 50 species, 56 today. Well, about the papers, we have taxonomic reviews for Anhydra, Eptanthus, Neurolaena, and just one section of Calea with 12 species. And the most of the papers are about new species and flora. And recently, studies with new approaches, fruits anatomy, morphometry, palinotaxonomy, and nomenclatural synopsis are provided. One quote about fruits anatomy, we discover a different pattern of expected for us, because Neurolaena is closer to Eliante. So we expected the pattern, the Eliante pattern of deposition of phytomelanin, but no. The 4 species of Calea analyzed for us show the eupatory pattern of phytomelanin, so it's very curious. We need more samples for see this pattern in all the genera of the tribe. So I will show some photos about the recently described taxa. Callia parvifolia, a notable species, the first notable species described for Callia. Callia roqueana, in honor of Nadia Rock, and Callia subintegerrima. We provided a synopsis for Neurolaeneae 40 years after the first synopsis of Turner about Neurolaeneae, and we described one new species, Neurolaeneae curtipalia. Prusky in 2023 published the taxonomic classification, a new taxonomic classification of Callia based on morphology data, and Callia was segregated in 7 genera: Laceanthus, Podocallia, Tepui pappus, Lemasium, Meyeria, Tonalanthus, and Callia also. And we have when I say we understand Gustavo Hayden, Morgan Gostel, Genilson Reis, and Jim Nakajima. We have a paper accepted in the taxon, and I hope it will publish and soon. Is the first taxonomic classification for the tribe based on the molecular and morphological results. This is the first molecular analysis for the Neurolaeneae and the genera of Neurolaeneae. We made a extensive morphological sampling. We described more than 150 species, describing the morphology for understand the results. And we tested the Prusky hypothesis. So our classification has showed the taxonomic key for the genera and the Callia subgenera, 4 subtribes, and diagnosis and occurrence for 4 subtribes, 6 genera, 8 subgenera, sections, and series. So this is the tree. The first very interesting result is Stauroclamis inserted the inside of Callia. Stauroclamis, if you remember, Robinson proposes as a part of Neurolaeneae, but after Panero, Funk proposed Stauroclamis in Coreopside. Now with the phylogeny, the first phylogeny for samples of Stauroclamis is nested in Neurolaeneae, close to Grimaniella. And the support is very good for this clades 100. For Callia, it's 100, so Callia is monophyletic. And not a 6 genera different. So in the red circles or red squares, you see general types of the hypothesis of Rusk. And if you see this name, Meyeria, you will see Meyeria, one species of Meyeria in this subgenus, Monocephalus, but also in this subgenus, Lemasium, more species of Meyeria in Nervosi, in Meyeria, and in Coronopifoli. So all the genera proposed for Prusky are not monophyletic. So when you see Callia is monophyletic, so we propose retain the names in the genus Callia and not segregating 8 different genera. So the great novelty for the tribe is a presence of Stauroclamis. Stauroclamis has just one species, is endemic from Savannah Cerrado in Brazil. And I don't know if anyone know, please say to me, but is the unique Asteraceae I know with a flattened involucre, and it's very beautiful, but I just know in the herbarium. I don't know in the field. In the future, I hope so. No. So about me, I'm a scientist with a disability. I have a Charcot-Marie tooth disease. This is a neurodegenerative disease. I lost the protective sheet in my nerves. out in my body, and this cause pain and less mobility, and I use orthosis for that, and deformities in the foot and in the legs mainly. So my first years as a scientist in the graduation, I made field trips for 2 years, but this caused a lot of pain. So it was recommended, please don't do that, don't do that. And I stopped my field trips in the graduation. This chair is very common in universities of Brazil, and it caused for me very, very uh, bad scenarios in the class. And I started see the class laying down on the floor because the chair is not appropriated for me. So I suffer a lot of ableism in my life, of course, and unhappily in academy also. So I will cite some phrases I listen or I read. One professor mine say, reading my project say, judging my project say, although I know the reasons for not doing a field work, this limits and diminish the value of the project. And another judgment say unviable project in my doctorate due to his limitation to not the limitation understand I don't I don't field trips. Sorry, this subject in this phrase is hard for me sometimes. I listen also. A good taxonomist personally knows the plant in the nature in the field. So with this phrase, I listen the phrase, this phrase several times. So I'm not a good taxonomist for these people. Saying unexpected morphological structure in a class about pollen. Whoa, look how different this morphological structure in the pollen, and the only is the sample, say the student. In the teacher answered, every population has its cripples, right? And he laughed it. I'm not. In a recent application, I listen. Your disease is degenerative. Have you ever thought about give up? And what will the future be like when your disease progress and you will still working? How? Well, in the post-graduation, I deal with a lot of buildings not accessible for me and not appropriated for different body like mine. And here I'm very happy in this moment. I returned for the made field trips and with this monocycle is a Brazilian monocycle appropriated for person with disable or person with limitations in the locomotion. And here I am hugging the Calha myrtifolia, and I'm very happy with Calha christini in two different moments. So how I don't visit the plant in the field a lot of times. I develop strategies for know the plants in the field. So I made one guide about Calha and Neurolaeneae and send emails and send for everyone in Facebook and the iNaturalist. And I use the iNaturalist. Every Sunday, I identify Asteraceae from Brazil and Neurolaeneae for the world. And today I'm a top identifier, a Neurolaeneae species in the iNaturalist. And with this, I know my plants in through the photos, and I know the plants in the nature with this. I described recently Callia funkiana in honor of Vic Funky, Callia sicilifolia, and Callia glandiflora. I show here because these three photos I received after the publication for different researchers sent for me, oh, this is Callia. And I say, oh, this is the new species I recently described. It's very is emotional for me see the plants in the nature because I imagine it, I see the cicada, but I did not see the photos, so with this photo is more fun show about that. In the post-graduation, I visit the USA and I made a visit in Smithsonian. I did my molecular analysis at the Brit with Morgan Gostel. And now I will say thanks for all this institution provide me funding and support for develop my research since from my graduation. And thank you everyone for see this presentation. I'm sorry for my English, and I'm trying improve the level of my English. And I hope everyone enjoy it, learn about Neurolaeneae.
Thank you very much. Great talk. Yes. Yeah, and very wonderful plant group, and also a really important topic about ableism in academia. So thank you for sharing about that too. So yes, if people have questions, you can put them in the chat or unmute yourself. Christoph.
Okay, now another question. I hope you hear me.
Yeah.
Okay. Thank you for your interesting talk. So to my eyes, the members of the tribe look very diverse, especially the achenes.
Yeah.
And so I was wondering, is there a synapomorphy for the tribe? Is there something that they all have in common?
In the type cell, I believe not. Maybe we expected find the position of phytomelanin in common all the genera, but we not test this. So the pappus is very different because we have bristles, we have a lot of things. Today I'm generating the phylogenome, and we will test the morphological characters and evolution of that for discover. But very interesting question because it's very different and it's very hard understand sometimes what made if is possible define one apomorphy for the genus in the subcell.
So the support is mainly from molecular data then?
Yeah, but we have I will try show again because I stopped the share. Sorry. If you can see here, striated in the filaris, we discovered this is ducts, resin ducts in Callia and in other genera. So this is a morphological shared all the genera because Eptanthus has, Enidra has, and Neurolaeneae has. So one hypothesis is this is a good characters for how close to these species, the genera.
Thank you.
You're welcome.
Mm-hmm. Thank you very much, Vinicius. I think we should go on, in the interest of time, to our last speaker. So our last speaker is Brannan Cliver, who will present their genome and morphology work on Hawaiian Bidens.
Hi, everybody. Thank you so much for having me. Let me figure this out. Okay, does that look all right? Awesome. Okay, hi, I'm Brannan Cliver. I'm a PhD candidate in the Kapitcha Lab at Auburn University currently, but we're about to move to Clemson University. I get to work with all of these lovely people listed here on the Hawaiian Bidens radiation. And I'm excited to tell you a little bit about it today. So to walk you through what I'm going to chat about today, first I'm going to introduce the Hawaiian Bidens and evolutionary questions we have for them. Pappus variation across Bidens, the genus as a whole, and then intro the genomes we've selected, and then explain autopolyploidy and allopolyploidy and some issues and challenges with assembling either of those. So to begin, this is the Hawaiian Bidens. They're very closely related. They hybridize readily. But despite the fact that they can hybridize, they don't often in the wild, mostly due to being so morphologically and ecologically, mostly ecologically diverse. This is just a couple of the Hawaiian Bidens that are found across the Hawaiian Islands. And actually only E there is one that we'll be really focusing on today. And this diversity, along with ready hybridizations between these Biden species, kind of led us to have a couple questions about them. The ones I'm going to talk about today are, what is the genomic basis for the phenotypic diversity across the radiation? And then what genes are directly linked to morphological variation in specifically achenes? And the reason we focus on achenes is because that is how they become dispersed. So this picture is of a beautiful owl, but I personally love it because each of these tiny little things here is a Bidens achene. And so Bidens get their genus name from bi meaning two and den meaning teeth. And you can see that pretty clearly with these micrographs that Erica took. They disperse via animals. We can get pretty hooked on Bidens in the greenhouse. But something that Erica noticed and we've noticed is that there's kind of a lot more diversity in the pappus number than the genus would lead you to believe. And so some more invasive Bidens that are found across the globe, like Pelosa and Bipinnata, have these really gnarly looking achenes with more than two pappus, really long pappus, whereas the Hawaiian Bidens that we're more focused on have little to no ons. Cosmoides doesn't really have any ons. Hawaiiensis has two, but they're a lot different than what the standard on is for the Bidens genus. And one Bidens that we're looking at a lot is this cultivated Bidens in the center here, compact yellow. Erica has been working on a population study of compact yellow, and she selfed the original cultivar and found that there's a lot of variation in the on number across her selfed population. So generally, she sees anywhere from 1 to 7 ons, and we're really interested in looking at this to try and better understand pappus development across the genus as a whole. So I'm going to switch gears just a little bit to explain that we are super interested in having genomic resources across this radiation. And so we've selected 6 genomes that we're super interested in and planning to assemble. One of them, the very first one we've kind of talked about a little with compact yellow, is Ferrulifolia. It's about as far out as you can get from the actual Hawaiian Bidens radiation, but it makes an excellent outgroup. Next to that is Bidens andicola, then Bidens pilosa, and finally we're getting into the Hawaiian Bidens themselves with Bidens cosmoides, Bidens hawaiiensis, and Bidens forbesii. So Ferrulifolia is actually a, compact yellow is actually a Bidens Ferrulifolia hybrid. It's cultivated species, it's diploid, but because of those hybridizations, we kind of hypothesize that there's 3 different parents for compact yellow. But because of that, there's a lot of heterozygosity in the genome itself, and we get to a really interesting study with the pappus, as I mentioned before. And I have actually already assembled this genome, which is why I know it's so heterozygous, because it was a little complicated to assemble, but it's done and I'm really proud of it. There's at least 5 really large structural inversions between chromosomes of haplotype 1 and haplotype 2, which is really interesting. We have a couple of genes of interest, not specifically for pappus development, but that are in those inversions. So it makes for a really interesting, just comparison. And yeah. So that's the first genome we've assembled. The next one is Bidens andicola. This is a diploid wild species, and we also hypothesize that it might be one of the parents of compact yellow. There's some evidence that we have that it's pretty closely related to compact yellow. And it's, I think I already mentioned, it's also diploid, which is important because from here on out, they're not diploid. So it'll be a little more difficult to assemble, but I'm also planning to use andicola to teach others within the study group how to assemble genomes. So I'm really excited about that. It's still not a Hawaiian Bidens, it's from the continents. And then we have Bidens pilosa, which if you were looking at the pappus differentiation across, not differentiation, pappus differences across Bidens, it was one of the invasive Bidens. But it's also hypothesized to be the origin of the Hawaiian Bidens radiation. It's also hexaploid, so it's kind of a big jump from andicola to pilosa, but I'm pretty excited about it. So after pilosa, we have the 3 Hawaiian species, Hawaiiensis, forbesii, and cosmoides. And yeah, basically just trying to gain a better understanding of how they evolved the way they did through these genome assemblies. So we have 4 hexaploid genomes, pilosa, cosmoides, hawaiiensis, and forbesii. And one thing about polyploid genomes is that there's two different kinds, autopolyploid versus allopolyploid. Autopolyploid are whole genome duplications that resulted from the same species, whereas allopolyploid resulted from a hybridization between two different species. And each of these have their own challenges when assembling, but allopolyploidy is definitely a little simpler to assemble than autopolyploidy. There's a couple different ways that I have seen people do it. One thing that we've actually already used for compact yellow, which is not a polyploid, but it had different enough chromosomes that we could use a Kamer-based subgenome phaser for it, which is how we kind of figured out that Andicula might be one of the hybrid parents. But you can use that for allopolyploids. Also, people have been using long-read Hi-Fi and Hi-C data. It just takes a lot more data than a diploid species would. People have also been pairing those two types of data with ultra-long-read data to kind of phase those subgenomes a little easier. Autopolyploids, on the other hand, are a little more difficult just because those subgenomes are so similar to one another. It can be hard to phase them into their own chromosomes and haplotypes. So some people will use both parental DNA data to assemble an autopolyploid, and you can do similar with the progeny of that individual that you're assembling a genome for. And both of these things can be paired with highly accurate ultra-long reads. The papers have specified highly accurate just because you don't want to accidentally purge any like SNPs or anything that may exist. So in thinking about this, we have kind of decided to go about it. There's, we're not totally sure what kind of polyploidy our genomes are. There's a paper that suggests ALBA, a related species, is allopolyploid, I think. So we're leaning that way, but the way that we've decided to assemble these genomes should work either way. And that is to use the hybridization of these species to kind of get data that we can use and align to the genomes. So basically crossing species together and then extracting DNA from those crosses and aligning that data to whatever assembly we're trying to that has one of those parents to essentially phase the genomes into the proper haplotypes. We have, as of now, we have just sent data off for Andicula, our second diploid species. We have DNA for Pelosa to give this a try, but we haven't sent it off for sequencing yet because we are in the process of moving. But hopefully in the fall, we'll be able to test and see if this works out. We're also waiting for our plants to flower so that we can begin those crosses. But yeah, that is all I have. I want to say thank you to the Capitulab and our collaborators at University of Hawaii at Manoa and University of Wisconsin-Madison. With that, I would love to take any questions y'all might have.
Thank you for a very nice talk. So yes, as always, people can unmute or write questions in the chat. So I have a question.
Yeah.
And that is, So do you think, so it seems like Hawaiian Bidens had just one origin, right?
Mhm.
And so do
I
you
think so.
Okay. Yeah, from your tree it looked like me. So are they all going to be allopolyploid or all autoploid, or was there enough hybridization that it could be different?
I think what we expect is that they would all be the same type of ploidy, whatever that polyploidy, whatever that may end up being. That was my assumption anyways.
Sounds good, but I guess you will find out.
Yeah.
Yes.
Brennan, I have one question. Have you ever encountered any species of cosmos?
Of cosmos?
Because cosmos is a sister group of Bidens. So many
Yes.
times what happens, it is treated as the species of Bidens, and many times it is treated as the separate genus. So just
Yeah.
for curiosity, I'm asking that have you ever encountered any species of cosmos?
I haven't when looking specifically at Biden's, but yeah, I don't know for certain.
Okay, no issue, because many times, delimiting both the genus are very difficult, because
Yeah.
you have explained about the purpose of Bidens.
Yeah.
So many times it happens that in Cosmos also these same situations occur. So it is quite difficult to delimit. So okay, nice work.
Thank you.
Okay, well, let us thank all three of our speakers again for three really interesting talks. And if people have other questions for any of the three speakers, you can be welcome to ask them. Okay. Yes, very, very interesting. So thank you all for telling us about your research. It's gives us all a lot to think about and introduces us to just some nice plant groups.